Hello and welcome to BioRules video lessons. Last class, we took a look on the phospholipid structure. So you have a bilayer of phospholipids that looks like this. No, head, tail, head. The tails are escaping from water. They are running away from water. The water is outside and inside over here. And the tails then run away from water, keeps the membrane together. The heads protect the tails. So beautifully, nicely, that's how the phospholipid bilayer is formed. We know this here very well now. Okay, you have their hydrophilic heads, we have the hydrophobic tails, beautifully, nicely, that's it. However, membranes contain many proteins, many proteins. And then the membrane is not only a phospholipid bilayer. Phospholipid bilayer is the basic structure over there, is what makes the membrane, but actually the proteins are going to be very important, the function, the structure, and all over the place. So where are these proteins? This image here, that actually I'm going to show you there, so the first transmission electron micrographs of membranes, they show this dark light, dark bands pattern, which led two scientists called Davidson Danieli, which I like to call them D&D, to think that maybe the phospholipid bilayer is in the middle over here, keeping the membrane structure together, and maybe the proteins, they are outside, floating here, one side, outside, and from this. So basically what he thought was that the proteins are here, in the outside and inside, making a layer of proteins with phospholipid bilayer there in the middle. So this is the Davidson-Danieli's model, okay? Proteins outside the bilayer, glued to the phospholipid bilayer, to the phosphate heads. And this is how scientists thought that the membranes was about in how they dealt with the proteins, okay? But there's a however. The membrane proteins, they are not entirely hydrophilic. For them to be totally outside of the membrane, like the Davidson-Danieli model proposed, they would have to be totally hydrophilic because they would be all in water. And the point is that they are not. They are, many of them, when I say they, I say most of the membrane proteins, they are going to be amphipathic. That means they contain hydrophilic and hydrophobic parts. So what? So what that if they have hydrophobic parts, then this hydrophobic part should be inside the phospholipid bilayer. So hydrophobic, I'm going to be painting here in orange, this part here, hydrophobic in contact with the tails. They are inside the bilayer. And hydrophilic over here, outside, in contact with water, out there and here. So this part here in contact with the heads and with the water are the hydrophilic parts. So if the phospholipid bilayer with the membrane proteins, if the membrane proteins are amphipathic, so they might be distributed in the same way that phospholipids are going to be distributed there. Who proposed this is two guys called Singer and Nicholson, making the Singer-Nicholson model, also known nowadays as the guys that were right. So proteins, they are embedded in the phospholipid bilayer. So they are now in the phospholipid bilayer, not just floating on the surface. This is the model that we have nowadays. What is interesting, we're going to be discussing this a little bit in class, is that sometimes we have a model that's wrong. But being wrong is not a problem for science, because sometimes, actually most times, when a model is wrong, it helps for scientists to preview other models, refine the model, to try to have a better understanding of how things are. So the Davidson-Danieli model actually is not an embarrassment for science. It's actually a model, knowing that you have proteins over there. They just got it wrong where the proteins actually were. Okay, Singer-Nicholson went a little bit further, knowing that the membrane proteins are not just there on the outside, they're actually happening all through the membrane itself. Okay, so just to show you the idea, this would be the idea of a membrane protein. So it's embedded in the phospholipid bilayer. In this here is showing going all the way through. Okay, but the point is, how does it work? I showed you before. If you have hydrophilic parts and hydrophobic parts, then we can have a protein with hydrophilic part and hydrophobic parts. So the blue is the hydrophilic, the yellow here is the hydrophobic. So you have hydrophilic, hydrophobic parts, and then this protein can be in the phospholipid layer happily and nicely and working, because the hydrophobic part stays inside the hydrophilic tails, in contact with hydrophilic tails, and the hydrophilic part stays outside, pointing to the water. Okay, so this is the idea here. No, they have their membrane proteins being amphipathic. No, so the hydrophilic in blue, the hydrophobic in yellow, and they stay in both mediums because they are hydrophilic, hydrophobic. Okay. Later, when you study biochemistry, you will learn a little bit more about proteins, a little bit more about hydrophilic amino acids, hydrophobic amino acids, how all this is possible, so it will make a little bit more sense later. For the moment, just get the idea that these proteins, they are amphipathic, they have hydrophilic part, hydrophobic part, and they stay in there. A consequence of this, look, is that we have models like this, and it's interesting that, okay, this molecule here, this one over here, it's quite obvious. Now you understand. It has a hydrophilic part here in contact with water. We have a hydrophobic part here in contact with the fatty part of the phospholipid, and hydrophilic back here again, in contact with water again. This is quite obvious. But what about this other molecule over here? What about this protein there? What happens here is that there's a part here in contact with water, okay, happy, outside contact with water. There's a part here in contact with the phospholipid hydrophobic part. And this is working as well, because this protein cannot pop out. Because if it try to pop out here, the hydrophobic part will get in contact with water, so it doesn't. And also it cannot pop in, because if it pops in, the hydrophilic part will get in contact with the hydrophobic tails, and they also cannot go in there. So the proteins can go all the way through and through, but they also can be kind of peripheric, just around there, being there and doing its stuff. Okay, so there it goes, lipid, hydrophobic, the tail. This one is called an integral protein because it goes through and through, and that other one is called a peripheric protein because it stays in the periphery, it stays in one side of the phospholipid bilayer. Okay, some more images for you. We have here more integral proteins. Let me mark this in red here. Look, more integral proteins, integral proteins. This one I don't know if it's integral because it's far away. No, this one looks like integral because I see the top and the bottom. And we have carbohydrate chains here. No, that some proteins have some carbohydrates, some sugar markers, sometimes are called a glycocalyx, that are visible over there. Here you have the carbohydrate group in glycoproteins, in glycocalyx. And what we have here, an integral protein, we have here peripheral protein. And one more thing that, it's over here, is this guy over there, cholesterol. Cholesterol is a fat, and fats, they stay here inside the membrane because this cholesterol that's present in animal cells, not plant cells, okay, cholesterol helps the animal cells to change its fluidity. Okay, look here, we have more cholesterol over there, and with having cholesterol or not having cholesterol, the membrane becomes more or less fluid. And that's important for animals that don't have cell walls, because they need to have a certain determined fluidity for the cell membrane. Okay, in this image again we see some integral proteins like this one here that actually has a hole inside so things can pass through. It's called a channel. Another one here with holes inside, a channel. This one is a glycoprotein. It has a sugar part and a protein part that anchors it over there. So many of stuff making this phospholipid bilayer. Look here again, glycoprotein. We have a glycolipid, which it's a sugar glued to a cholesterol to stay in place. Cholesterols here to change the fluidity. We have integral proteins over there. We have peripheral proteins over there. Here you have a peripheral protein that is only hydrophilic because it glues to another protein. There's an integral one. So, beautiful view of our fluid mosaic model. And here, a drawing that I did myself to show you lots of phospholipids making a phospholipid bilayer. I'm leaving some spaces there to draw to you some proteins as soon as I finish here. So I will draw a bunch of hydrophilic heads in blue, hydrophobic tails in yellow, and then I'll put the little proteins around. And that is it. Here I have proteins, 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 including a glycoprotein over there, cholesterol, so all is there. So we have phospholipid bilayer. Phospholipid bilayer is made by hydrophilic head, hydrophobic tail, and another hydrophilic head on the other side, okay? I have a glycoprotein, which has the glyco part, the sugar part, with the protein part over here, integral protein. I have, no, that's the carbohydrate. I have cholesterol, which changes the fluidity of the membrane, embedded in animal proteins. We have an integral channel. It's a channel because it has a hollow inside, so things can pass, but it's an integral protein. It's a channel, but integral protein. There we have a peripheral protein, which is glued to another integral protein on the side. So this here is, now we have there another integral protein, transmembrane, because it goes from side to side. So we have our phospholipid bilayer with proteins, which makes a fluid, because it moves around, mosaic. And this idea on fluid mosaic is very cool, because if we put a protein, we'll see that the proteins, they can move from side to side. So proteins, they can move freely from side to side because no, they are there, but they cannot jump out or in or flip. So a protein cannot jump to the side because the hydrophilic part cannot go through the hydrophobic inside here, so they cannot flip. And they cannot pop out because the hydrophobic part of the protein over here cannot face the water outside. So proteins, they move sideways, what makes them fluid, but they stay in place, not flipping, not popping out, not popping in. So this is the fluid mosaic molecule, okay, of membranes. The Singer-Nicholson model that shows us is the fluid mosaic model, okay? If you want to see more about this, when I say if you want, it's kind of an euphemism. Please go ahead, get a cell phone, take a look on this link and see this video. We saw it before for the eukaryotes, and now you're going to be seeing a vesicle made of membrane over there carried by a protein. You're going to see here, this is the hydrophilic heads facing outside with proteins. This is a protein raft moving outside. And if we see in a cross section, we have here heads, tails, heads. We have cholesterol over here, many of them. And we have, look, an integral protein. We have hydrophilic, hydrophobic, and hydrophilic back here again. So I strongly advise you take a look on this video to have a better idea on how the membrane works and how beautiful it is. I hope you have enjoyed this class. Do the quiz now, have fun with it, and I'll see you in class so you can have a better discussion of the fluid mosaic model.